Okay. Hello. Can you hear me? That's the question. Can you all hear me? Yes, brilliant. Okay, that's great to hear. Okay, let's just get this camera sorted out a little bit. There we go, that's a bit better. Try not to chop my head off. Excellent, okay. Right, well, sorry, there was a slight delay there. It's just that we haven't done them back to back before, so it's the signing in process. So welcome. Please let me know if, how many of you have tried some of this Snap Revise before? Hello, how are you? I'm good, are you good? Excellent. So I hope everybody's looking forward to going back to school next week, if you're in the UK. So, and hopefully you're all keeping well and you are getting used to the lateral flow, the idea of the lateral flow test. We're just waiting for a few more people to join the web class. So just while we wait to make sure that we've got everybody into the web class. Oh, second lesson. Well, brilliant. Excellent. Have an exam on stem cells tomorrow. My word, this couldn't have been a better time for you, could it? Excellent. Yes, finally. Okay, finally back to school. I know a lot of my students, basically the lateral test works biologically. Well, that's kind of for another day. I can't do that today while we're doing about stem cells. But yes, I think that's something that would be interesting. We could, if you send a message in to Snap Revise, I'm sure we can get something sorted out. You can't wait. I'm doing triple science AQA, so confusing. Don't worry, we will sort you out, I am sure. All right, so we will be getting started soon. So do say hello to each other. And let me just check the best way for communication. Yeah, so let's have a look. So every week, go to each noise, it's coming higher. How can I find out? So try Instagram. Instagram looks like it's the best way to get hold of us. So try sending a message on Instagram to see if there's something that you need covering. All right. Is this AQA? Well, it's a mixture. It could be AQA, OCR, EdXL. So we try to cover all of the exam boards for you. A lot of them are very similar. Yeah. Sorry, I'm not sure. It's all the exam boards, I think, right? In picking my options, but think I will do triple science. Well, it's worth hanging on, seeing how you get on. Triple science, yeah. Right. So I think we should start soon. And what I'll do is I will look at sharing my screen with you. Okay, so let's get this started on stem cells. So I'm going to share my screen with you all now. And can you see my screen all right? Can you see the start of a slideshow? Okay. So hopefully you can see that. Yeah, brilliant. Good, good. So you can see the screen, and this is the start of the stem cells web class. Do try answering questions as we go through. I'm going to be asking you questions as we go through. So do ask questions, answer questions, get fully integrated in it, because that's what you're going to get the most out of it, aren't you? So welcome to you all. We're going to start this web class on stem cells today. And the more participation, the better. First of all, let's introduce myself, shall we? So you know who's actually teaching you today. Well, I'm Philippa. I am head of biology at my school where I teach full-time. I have a bachelor's degree in animal science and a PGCE in biology and games, both of which I read at the University of Leeds. I've been teaching and tutoring for over 25 years. I've also been an examiner and have written for various academic publishers. So I do hope my experience is going to be helpful to you. I love my job and I hope that my enthusiasm for the subject may at least partly rub off on you. Okay. So just before I start the web class on stem cells, I just want to tell you about GCSE tutoring, a new platform which Snap Revise is just launching. So Snap Revise provides these sessions with running on YouTube, but there is an extensive platform full of resources for you. And they're tailored to GCSEs and A-levels. So we have lots of new additions, including the GCSE tutoring platform, and that's covering the full curriculum. So we're covering your AQA, Edexcel, OCR, and we're going to, the classes each week work logically through the curriculum, fine-tuning all the skills needed to achieve your potential in your exams and assessments. There are high quality handouts for you of the PowerPoint slides, and they're going to help you with your notes and learning. Recordings of previous lessons are available in case you want to re-watch them, to go back over a particular topic. I know that I find that if I was learning something, then there's certain topics that you just want to go back over. And there's also one-to-one support where you can ask questions about specific areas you're not sure about. And so that should help you with your learning. Okay. If we have time at the end, I will quickly show you that on the Snap Revise website. Okay. Another thing that you might be interested in, because everybody likes to have a freebie, if you're interested in the tutoring platform that I was telling you about, then there's a free 2-week trial. Just sign up on the website. You enter this code, tutor14, at the checkout, and you can claim the trial. You've got nothing to lose and hopefully a lot of knowledge to gain. So let's see. So it does not let you enter a code. Right, that will be sorted for you. Hopefully somebody will reply to you for that, tutor14, to get your 2-week free trial. So let's start our lesson on stem cells. So the learning objectives for this lesson are to be able to describe the function of stem cells in humans and the potential for their therapeutic use. In other words, how can they be used to treat disease? Be able to discuss the potential benefits and risks associated with the use of stem cells in medicine, and also to describe the function of stem cells in plants and the potential for plant cloning. I won't be covering cancer in this particular web class. Here are the specification points. You were asking about what exam boards this covers and whether it covers just AQA. Well, it doesn't just cover AQA. This is the list of specification points for the exam boards. I'm not going to go through them all with you because it would get a little bit boring and tedious, but the specification booklets are available for you to download on the relevant exam board websites. Okay, so there's the OCR, there's the Edexcel. It's a really good idea to download these specifications as it's a good habit to get into. You should be looking at your spec as you go through your course and during your revision to check off points as you cover them and to make sure you do not forget to go over anything. Excellent. I always get amazed about how many students don't actually look at the specification. And it is really important. It should help you with your revision. Okay. So let's look at, let's get the pen up. Okay. And let's look at what we already know. Can you tell me why cells divide by mitosis? So what can we get for why do cells need to divide by mitosis? To repair and growth, great. For growth and repair, to replace, to repair and grow. Brilliant, okay, so growth and repair. And development as well, development. I love it when my pencil froze, I'm afraid. Growth and development, replacing old or damaged cells. And what's special about mitosis compared to meiosis? What do we end up with at the end of mitosis? Are all the cells different? Are they the same? Do we know? Anybody got any ideas? Same daughter cells, brilliant. So they form genetically identical daughter cells. Good use of the word daughter cell there. Brilliant. So looking at the next question then, why do cells differentiate? So this might be taking you back to quite near the beginning of the course when you talked about possibly specialized cells, and you will have looked at various different specialized cells. So why do cells differentiate? Any idea why they would differentiate? In other words, they form specialized cells. So when a cell differentiates, it forms a specialized cell. Why do we need this? To perform specific functions. Excellent, Shana. That's really good. Good. To carry out functions. Brilliant. And purposes. Good. Well done. Ellis, is that? So to carry out particular functions. Because cells specialize and acquire different subcellular structures. So the cells specialize and acquire different subcellular structures, such as the axons of neurons, or the villi in the ileum, the microvilli on the villus cells, okay? And now obviously for cells to work, they can't just work individually, can they? What do they do? You don't just have individual cells all around the body just working by themselves. We have this sort of organization, don't we? Can anybody remember about, they're usually an hour. So the sessions are usually an hour. So when we have lots of similar cells working together, similar cells working together, what is that called when they're all working together? Can anybody tell me? Tissue, brilliant. Okay. And those build up into Tissues work together and build up into, anybody got any ideas? So we go cells, tissues, organs, organism, yes, organs, organ system. So it could be that we've got muscle tissue building up into heart, which builds up into the circulatory system. Into organisms. Okay, well done. That's a good start, a good warm up to get us going. So these cells have to come from somewhere, and they come from other cells by mitosis, so that we're making these cells which are genetically identical daughter cells. And the cells that these cells come from before they can differentiate are our stem cells. So now let's look at human stem cells. So we'll start with the human ones. We will talk about the plant ones as well. But what different types of human stem cells are there? So what is a stem cell? I've kind of hinted at it already. What are stem cells? Anybody got any idea? So we've kind of talked undifferentiated cells. Brilliant. So what are stem cells? They are undifferentiated cells. So they divide, they form these identical daughter cells. We get these identical cells which have not got any specialization. They're undifferentiated at this stage. They haven't got any specializations yet. And they're there to keep replacing cells, because some cells just keep coming off the body all the time, like skin cells. That's what some of the dust is in the air. Our old skin cells, they have to keep being replaced, don't they? So what can stem cells divide into? Oh, good. Well done. Somebody's just answered what I was asking earlier about the different types of stem cells. Okay. Specialized cells. So they're going to eventually produce specialized cells, aren't they? So what can they can go into, they self-renew and produce many more stem cells. They can then specialize into things like red blood cells. Good. Okay. So they self-renew and produce more stem cells. Because we have to keep replacing those stem cells as we're being used. Okay. Meristems in plants, well done. Meristems in plants are dividing areas in plants. So those are the ones that are in plants, not in animals. And then, like you said, they then differentiate into new cell types. Good. Such as your red blood cells. In other words, they go into specialized cells. Like you can see at the side here with our nerve cell here and our red blood cells and our muscle cell. And that looks like ciliated epithelial cell with the mucus on top from the goblet cells. So when they differentiate into specialized cells, they are now known as unipotent. In other words, they can't divide. They can't divide to form any other type of cell now. So once they've differentiated into a specialized cell, they then stay as that particular type of cell. So they're known as unipotent. They cannot divide. Uni being one, so unipotent, they can't divide into any other type of cell now. All right. They also cannot change back into a stem cell, not naturally anyway. Okay. Mammals have two types of stem cells, and it was already mentioned in the chat there, well done, that the two types of stem cells in humans are embryonic stem cells and adult stem cells. So embryonic stem cells, funny enough, come from the embryo. What time does this lesson end? It finishes at 7 o'clock, okay? So the mammals have two types of stem cells, embryonic stem cells and adult stem cells. And the embryonic stem cells are the ones that come from the embryo. So they've got to be able to divide into any type of body cell, all right? So they can divide into any of the 220 or so different types of body cells. So they're known as either pluripotent or totipotent. Pluripotent meaning any type of body cell. Totipotent also means that they can divide, they can make placental cells. What does epithelial mean? That's a surface. That's a surface cell. So epithelium is like we have epithelial cells of the skin cell. We've got epithelial cells lining our trachea. So epithelial cells sort of line areas and cover areas. All right. So hi from Barbados. Excellent. Welcome, Shem from Barbados. Right. So adult stem cells are different to embryonic stem cells because embryonic stem cells can divide into any type of cell in the body. Adult stem cells can't. So they're known as multipotent, which means that they are more restricted in what they can divide into. So the pluripotent means they can divide into any type of body cell. So those are the embryonic ones. And multipotent is adult. So I always think that plural doesn't have an ending to it. Multi just means many, doesn't it? Like a multi-story car park. So multipotent, the adult ones, it can grow into a reasonable number. So you have blood stem cells, which can change into the various different types of different blood cells. But embryonic ones can differentiate into any of the body cells, or you can call it totipotent if they can also form the placental cells. Okay, so that's the introduction to our stem cells. So let's have a look at embryonic stem cells then. So if we're going to talk about embryonic stem cells, you need to know what an embryo is. What do you actually think an embryo is? When does it form? What can we come up with that? So an embryo, what do you think an embryo is? An undeveloped cell, a zygote, a fertilized egg. Good, we're getting some good ones here. Okay, so a fertilized egg forms a zygote. So when a sperm and an egg come together, then we get a zygote, and that's the fusing of their nuclei. And then after that, it then divides into a ball of cells, basically. So here is the, this is in the fallopian tubes at the top here. And that is, it would have started as one cell. So the zygote is one cell, and that zygote then divides by mitosis, forming a ball of cells. I don't know whether anybody knows what the ball of cells is called. It begins with a B. You might not have covered this yet, depending. So male and female gamete fuse, you're all getting really good ideas here. When a zygote undergoes mitosis, yeah, brilliant. So the fetus comes afterwards. So what we have is we have blastocyst here, a blastocyst, okay. Cyst is cell. So blastocyst is basically a bunch of cells. So what happens is when the sperm and the egg fuse together, then they form the zygote. The zygote then divides by mitosis and forms this ball of cells called a blastocyst, which then moves down the fallopian tube to the uterus. Well done, good, blastocyst. Good, just careful with the spelling, CYST for a cell. So blastocyst, that's brilliant. And then the blastocyst will embed itself into the wall of the uterus, so the lining of the uterus. And so then it starts to form an embryo. So an embryo is an unborn offspring during early development. All right. So at fertilization, we have this forming of a zygote, and now we've got the normal number of chromosomes, the diploid number of chromosomes, divides by mitosis to form this blastocyst, which is a ball of identical cells. They haven't specialized yet. And then when they hit the uterus, they then start to specialize. And now we're starting to form the embryo, which is the unborn offspring, which is between 1 and 9 weeks old. All right. So it is an unborn offspring during early development. So between 1 and 9 weeks old, all right, formed after fertilization. Brilliant, well done. But how does this small ball of cells, the blastocyst, then form an entire organism? Okay, so within a few days of fertilization, the cells divide rapidly by mitosis. And then, like I said, they move down the fallopian tube, they hit the uterus, and then once they hit the uterus, they then embed into the uterus lining, and then they start to differentiate, which once they've differentiated, they can't change back into stem cells or turn into any other cell types. They are now known as unipotent. Okay. We, at some stage, I'm sure we'll be doing meiosis. We will be covering the whole course at some stage. So embryonic stem cells can develop into, what can they develop into? Can you remember what that pluripotent means that I mentioned? So with the pluripotent, it means it can differentiate into what? Can anybody remember? Develop into many different specialized cells. Yeah, any type of body cell. Good. Any kind of cell. Yes, good. Without, not the placenta, because that's the totipotent. Well done. You are doing, yeah, you're really picking it up. So any specialized cell type found in adults. So you can call them pluripotent or totipotent. Okay, good, well done. So now let's look at the adult stem cells, shall we? Where do you think adult stem cells are found? Well, they're found in lots of different areas of the body, including, have a look at the diagram there. That should be able to help you get at least one of them. Totipotent, by the way, was it can divide into any body cell and also into placental cells, which aren't actually part of the embryo. Okay, so those are totipotent, but the placenta has to form somehow. Okay, brilliant. Okay, so you've come up with one, brilliant there, Sarah. So that's the brain, that's good. What else have we got? Bone marrow, okay, brilliant. So we have bone marrow. I mentioned another one today about dust in the air. Anybody remember what one of the things is that forms dust in the air? Okay. Bone marrow, skin cells, brilliant. Skin cells, all right. Brain, somebody came up with right at the beginning. We've already had that. Umbilical cord, somebody's come up with. Good, umbilical cord. There's also a very large organ in the body, which carries out loads of different things for us. I don't like the taste of it though. Okay. So the liver is another one, and intestines. Yes, we will be mentioning briefly about the ethical issues of the use of stem cells, of embryonic stem cells. We're not going to get into a long discussion about it, but we will mention it. Yeah, liver, good, well done. So adult stem cells can only develop into, so they're known as multipotent, as they can only differentiate into, what have we got here? So the bone marrow cells, what can they develop into? Anybody got any idea? What do those bone marrow cells? The diagram here is giving you a clue. Anybody got any idea on those? Oh, thank you very much, Sarah. Red blood cells, brilliant. You can spot the red blood cells. So we've got, so we have got, they divide into a limited, they develop into a limited number of cells found in that location. So this one, as an example, the bone marrow, we've got three main different types here. We've got the red blood cells. We've got, these are the phagocytes, which help fight disease. And then we've got the very important lymphocytes here, your T and B lymphocytes, very important in vaccinations, okay, which is very current at the moment, isn't it? So adult stem cells, can anybody remember what potent they are? It's not pluripotent. Can anybody remember what the adult stem cells were? They were something potent, which means they can produce various different things. Brilliant, multipotent, excellent, multipotent. So they're multipotent, and they're used for repair of tissue and replacement. We don't want to run out of any of these cells. They're really important. Brilliant. I'm really glad people are getting the hang of this now with the potent, the multipotent, used for repair and replacement, because we would have a bit of a disaster if we ran out of red blood cells. We wouldn't get enough oxygen around the body. We wouldn't be able to respire, and it wouldn't be a very good place to be. So the adult stem cells, like I said, are known as multipotent, and they differentiate into limited numbers of different cells, as they can only be found in specific parts of the body. So they form after the embryonic stem cells divide. They can only divide into the limited number of cells found in that location. For example, our bone marrow. So when the body is injured, we need to repair it. When cells age or are destroyed in places of high turnover, like our skin, then we need to replace them. Some animals can regenerate whole parts of their body. All right. So lizards, for example, if they get trapped, if they get caught by their tail, they can lose their tail and then they can replace it at a later stage. Same thing with starfish and their limbs. They can regenerate. And there's been a lot of research into this for medical usage. All right. So despite their name, adult stem cells, they can be found in children too. All right. And it is, from research, we've found that adult stem cells can actually be more versatile than they previously thought, but they have to be treated very carefully. All right. And no, I don't think so with the heart. The stem cells are producing stem cells which can then possibly be injected into the heart to replace damaged tissue in the heart. Okay, so how are we feeling so far about this part? Do you think you're ready for some exam questions? Have you got any questions at this stage, or would you like to go on to an exam question to see the sort of thing they can come up with? Yay, brilliant. Okay, good. There's lots of ones coming out there. Great stuff. So let's look at this exam question. This is from the Edexcel board. It doesn't matter which one we take them from really, but this is from Edexcel. And it's suggest how stem cells produce new tissue. All right. So look at this. There's 2 marks for this. So we need to give 2 individual points. So suggest how stem cells produce new tissue. So first of all, if we're going to produce new tissue, what's tissue made up of? There are stem cells in the heart. Brilliant. Well done. That's what I like to see, people actually looking it up and finding things out. Yeah, stem cells can differentiate into specialized cells. So the tissues are made up of cells. So first of all, these stem cells, if they're going to make a new tissue, they have to make new cells, don't they? All right. So how do they make those cells? By what process do they make the cells? We mentioned it right at the very beginning. Okay. So we mentioned it right at the beginning. They divide by mitosis. Brilliant. So the first thing you need to do is we're looking at a tissue, but a tissue is made up of cells. We need to find out how they make the cells. So stem cells divide by mitosis. So that will give you 1 mark. Okay. And then a lot of you have already got it. All right. How do they then make the new tissue? Well, these are specific cells making up this tissue. So now they need to differentiate, don't they? All right. And also differentiate into specialized cells. Well done. So that gives you your 2nd mark. Good. Okay, how are we doing? Right. So we need to mention about mitosis and differentiation, and that gets you your 2 marks. Right, let's have a look at the next one then. Now let's look at this one. This is a higher paper one. Adult stem cells make red blood cells. State 1 similarity between adult stem cells and embryonic stem cells. So what links them? What is the general rule of stem cells? They haven't gone through something yet. Okay, this one is Edexcel again, this one. We will be doing some others. We will be doing other examples, don't worry. They both can replace unhealthy cells. They can both specialize. So they haven't specialized yet, have they? Good. So state 1 similarity, both are unspecialized cells. Which differentiate at some stage into specialized cells. Okay, and then state 1 difference between adult stem cells and embryonic stem cells. So 1 difference. I think you can all manage this now. Adult stem, yes, they're multipotent, while embryonic are totipotent or pluripotent, whichever you want to use. So embryonic cells are pluripotent or totipotent. And adult stem cells are multipotent. So you have to, because we're looking at the difference, we have to say what both of them show, all right? So the embryonic stem cells are pluripotent or totipotent. Adult stem cells are multipotent. But once they've all differentiated, they all end up as unipotent. Good. Yes, this is the higher one. Well done. Yeah. All right. So now we've had a look at those stem cells. We can now look at the bit that people talk about, where the ethical side of it, but stem cells in medicine. So we're going to look at the difference between embryonic stem cells and adult stem cells and the way they can be used in medicine. Okay, it's very clever how stem cells, how they're developing the use of stem cells. There's still a lot of research to be done, but it is really good. So let's have a look at how the stem cells are gathered and used. Which type of stem cell is more useful and why? So let's have a look at this. Embryonic stem cells, we've just answered a question on this. Embryonic stem cells are pluripotent, adult stem cells are multipotent. So in theory, the embryonic stem cells are going to be more useful because they are pluripotent or totipotent. But does this actually always ring true? So when we have a look at them, we can look at the embryonic stem cells as they're pluripotent, totipotent, and they can be cloned and made to differentiate into most cell types. So how do we get these stem cells? Well, the embryonic stem cells can be used directly, as they can naturally divide into most types of body cell, that pluripotent, totipotent. And the best source of these cells is what makes it debatable. A lot of people discuss the ethical side of this, but they can be used directly from, does anybody know? A special treatment that some people have to pay a lot of money for a lot of the time. Yeah, okay. So IVF, in vitro fertilization. So the embryonic stem cells can be used directly from IVF. So the best source of these stem cells is from a 5-day-old embryo, often used from leftover embryos from IVF treatment. All right. So they can then just be cloned, grown in the lab, and then they can be treated to develop into different types of cells. But embryonic stem cells can also be generated by a process called therapeutic cloning, which is very, very clever. This has the advantage of providing cells with the same genetic makeup as the patient. All right. So yeah, they can be used. They're trying them out on cancer with transplant. So transplant, cancer, IVF. IVF stands for in vitro fertilization. Okay, so it's fertilization in the lab, what people used to call test tube babies at the beginning. All right, so they remove the eggs from the female, fertilize them with the sperm, and then they grow the embryos in the to implant into the recipient. Try to get the words out there. The recipient. All right. So therapeutic cloning, what they can do is they can actually take cells, such as a skin cell here, from the patient who needs to have some sort of treatment. And what they can do is they can actually take the nucleus out of it. So this is the nucleus. They remove the nucleus from that skin cell. And what they then do is they take an egg cell. So this is an egg cell, which they then enucleate, which means that they remove the nucleus from it. So now we've just got an empty egg cell. And what we can now do is we can place this nucleus from the patient here, who's not looking very well up here. We can take their nucleus and they can be put into an egg cell, which then means that egg cell can then be cloned to produce stem cells. All right, so they're taking the nucleus from a cell, like a skin cell, from the patient. The nucleus from this cell is removed and placed into an enucleated egg cell, an egg cell which has had its nucleus removed. So we're putting the genetic material into the egg. All right, so we're putting the genetic material into the egg. All right. The egg cell has been taken from a donor. This egg cell is then stimulated to divide so that stem cells are produced from it. And then these stem cells, which are either from the IVF, the embryo from the IVF, or from therapeutic cloning, can then be treated in particular ways to form our different types of cells, like our muscle cell and our nerve cell, and I think that's the pancreatic cell. All right. So uses of stem cells then. Both the stem cells made from the egg cell with the adult nucleus and the embryotic stem cells can then be cultured and treated to make them develop into the type of cell required, such as cardiac muscle to treat heart attacks, neuronal diseases, so produce new neurons, and also produce pancreatic cells to treat diabetes. So uses for stem cells are growing the cells and tissues. And why would we need to grow those cells and tissues? Okay, so why do we think we need to, can you remember why we might need to grow these tissues for these patients? Okay, to multiply them, good. To multiply them up so that they can grow cells and tissues to replace those diseased ones. Okay, so that's one reason why we want to grow stem cells. But there are actually two other reasons that a lot of people don't think about. All right, because we've got to get this, we have to find out about these processes in the first place, don't we? All right, so what other do you think to replace them, to recover, to repair? So that's all about the treating of the patient, but can anybody come up with any other ideas about why we might actually want stem cells, to grow stem cells? I read a very good book, The Immortal Life of Henrietta Lacks. It's a very interesting one about stem cells. Okay, cloning plants. Yes, we're going to be going on to that very soon, actually. And also, yes, well, we clone, yeah, bananas and foods. We're going to be getting on to that. But another reason is to actually study the differentiation of cells and disease so that we can actually help when there might be a genetic disease. Okay, so to learn how to treat the conditions and how to study, we want to study the differentiated cells to see how they work properly and how they actually differentiate, and to study the diseases. And then once we study the diseases, we might be able to grow to treat paralysis, good, to treat diabetes, but we also need to test drugs and medicines. And you can actually trial drugs and medicines on stem cells, which means that they can be tested on those before tested on actual people. So testing drugs and medicines. Okay. So using stem cells then, benefits and concerns. Let's see how we're doing. Crikey, we're running out of time here. So using stem cells, the benefits and the concerns of using them. So benefits, well, we've already done the treating disease, haven't we? And somebody just came up with paralysis as a good example. So they are treating disease, blindness, diabetes. Therapeutic cloning can be used to give cells which actually have their own genetic makeup. Does anybody know why that's would be really, really useful? Okay, treat paralysis. So likely to be, less likely to be rejected. Okay, no risk of rejection, well done. Right, and the next one, studying disease development. I do apologise for my writing. But obviously we have concerns, okay? There's ethical and religious concerns. About using embryos. And also, when we actually put stem cells into somebody, there is a risk of viral infection. Oh dear, that didn't come out very well. Infection transfer. I was trying to write too quickly. Risk of viral infection transfer. Okay. Yeah, immunosuppressants. So they are, yeah. Okay. Good. So moving on then, because we've gone through all of those. How confident are you feeling now? Have you got any questions for me? Are you ready to look at some more exam questions? Okay. Yay, good, brilliant. Okay, so let's move on then and have a look at these then. Suggest why it is ethically more acceptable to take stem cells from an umbilical cord instead of using stem cells from a 4-day-old embryo produced by in vitro fertilization. What do we think for this one? Anybody got any idea? So why do we think it's okay to use stem cells from an umbilical cord possibly after birth rather than from an embryo? People have ethical issues. Yep, good. So so we're not destroying the embryo's life. So destroying potential human life. Stem cells taken from a child's umbilical cord could be used to treat a condition later in that child's life. Give one advantage of using the child's own umbilical cord stem cells instead of using stem cells donated from another person. This is the same advantage as the therapeutic cloning. All right, yeah, not going to be a person, religious objections, a right to life, yes. Embryos have a right to life, that's always one thing. No rejection, good. So there's no danger of rejection. Well done. Why would it not be possible to treat a genetic disorder in a child using his own umbilical cord stem cells? I think the important thing here is it's a genetic disorder. So why would it not be able to, why couldn't we treat a genetic disorder in a child by using their own cells? Because it'd be using the same DNA. Fantastic. Yeah. The stem cells would have the same faulty gene. So that's important to read the question, isn't it? Okay. Right, very briefly then, because we haven't got all, it looks like we're coming to the end of the lesson. Just to quickly show you, the meristem is the multiplying area of the plant cell. So they also have plant stem cells, all right? So they have stem cells which are different to human cells because they have a really useful stem cell because they have the ability to specialize into any plant cell type throughout the plant's life. They don't get to adulthood and then stop, all right? So plant stem cells are different to human stem cells because they keep their ability to specialize. They don't have the difference between embryonic and adult stem cells. So they keep their ability to specialize into any plant cell type throughout life. Okay. Yeah, I think that's a good idea to do this bit in a drop-in session so that we don't have to rush it through. Yeah, what a great idea. You are very sensible. So I think what we will have to do is we will come to, it's because there were so many good answers. That was really good. So just to remind you, do go and have a look on the Snap Revise do have a look on the Snap Revise website for those free 2-week loads. All right, so my next lesson is on Wednesday, and I'm going to be looking at homeostasis in my next lesson. Okay, so next lesson is on Wednesday, and it's in year 11, homeostasis in the nervous system.